to our Glory Globe TV. Here's a breaking news. Yes, a groundbreaking study reveals how inherited genetic variants contribute to the development of rare blood cancer, offering insights for disease predictions and treatment strategies. Large-scale genetic analysis has helped researchers uncover the interplay between cancer-driving genetic mutation and inherited genetic variants in a rare type of blood cancer. Researchers from the Wellcome Sanger Institute, the University of Cambridge, and collaborators combined various comprehensive data sets to understand the impact of both cancer-driving spontaneous mutations and inherited genetic variation on the risks of developing myeloproliferative neoplasm, that is called MPN. The study published today, January 17, in Nature Genetics, describes how inherited genetic variants can influence whether a spontaneous mutation in a particular gene increases the risks of developing this rare blood cancer. This analysis has an impact on current clinical predictions of disease development in individuals. Further research is required to understand the biological mechanism behind how these inherited genetic variants influence the chances of developing rare blood cancer. In the future, this knowledge could be aid drug development and interventions that reduce the risks of disease. Myeloproliferative neoplasm, that is called MPNs, are a group of rare chronic blood cancers. There are around 4,000 cases of MPN in the UK each year. This occurs when the bone marrow overproduces blood cells, which can result in blood clots and bleeding. MPNs can also progress into other forms of blood cancer such as leukemia. In the population, there is a large amount of natural variation between individuals' blood cells, which can affect the amount of blood cells a person has and their particular traits. This is because multiple different genes can influence blood cells features in an individual. During routine blood tests, researchers take known information about these genes and analyzing the variation to give the genetic risk score, which is how likely that individual is to develop a disease over their lifetime. MPNs have been linked to random somatic mutations in certain genes, including in a gene called JAK2. However, mutated JAK2 is commonly found in the global population, and the vast majority of these individuals do not have or go on to develop MPN. Whilst previous studies have identified over a dozen associated inherited genetic variants that increase the risks of MPN, these studies insufficiently explain why most individuals in the population do not go on to develop MPN. This new study from Wellcome Sanger Institute of collaborators combined information on the known somatic driver mutation in MPN, inherited genetic variants, and genetic risk scores from individuals with MPN. They found that the inherited variants that cause natural blood cell variation in the population also impact whether a JAK2 somatic mutation will go on to cause MPN. They also found that individuals with an inherited risk of having a higher blood cell count could display MPN features in the absence of cancer-driving mutation, thus mimicking disease. Their study helps us understand how inherited DNA variation from person to person can interact with cancer-causing mutation to determine whether disease occurs in the first place and how this can alter the type of any subsequent disease that emerges. Their hope is that this information can be incorporated into future disease prediction efforts. Behind the report, Glory Globe.